Good evening to everyone.
Can you keep on presentation mode?
Okay.
Yeah, perfectly fine. You can start now.
Thank you. First of all, I am going to introduce myself. I am Dr. Sumaira Anjum, working as assistant professor in Kinnaird College for Women, University, Lahore. Today, I am going to present the topic related to the light tailoring impact of UVC light on biosynthesis, physiognomes, anticancerous, and biological activities of Morus Macroura-Mediated metallic and bimetallic nanoparticles. First of all, I would like to introduce briefly about the green synthesis of the nanoparticles, as the main aim of my study was the green synthesis of the nanoparticles, in which I have used the plant extract of a medicinally important locally inhabited plant, Morus-Mediated, commonly known as the king mulberries in Pakistan. In green synthesis, you can use various other sources of eco-friendly, like you can use microorganisms, you can use plants, and you can use biomolecules like proteins, lipids, and vitamins for the synthesis of the nanoparticles. In this study, I have used the plant extract for the synthesis of the nanoparticles, and the nanoparticles I have synthesized was the metallic, let's say like silver and zinc oxide nanoparticles, and also the bimetallic nanoparticle, in which these same entities like zinc oxide by silver nanoparticles, bimetallic was synthesized. And further, we have evaluated the different biomedical applications of these synthesized nanoparticles. This is what were the main objectives of this study. First, to synthesize the monometallic and bimetallic nanoparticles. In monometallic, silver and zinc, and bimetallic, silver zinc oxide nanoparticle were synthesized. Then we have performed the phytochemical analysis of the plant extract, and further, after the controlled synthesis of the metallic and bimetallic nanoparticles, we have evaluated the possible effect of UVC radiations on the synthesis, and obviously on the morphology, crystalline nature, and other physiognomes of these monometallic and bimetallic nanoparticles. To characterize the various physiochemical genomes, or how the UVC light impacted the different physiochemical properties of these monometallic and bimetallic nanoparticles, we have characterized these nanomaterials by various techniques. And after successful characterization, we have evaluated different biomedical applications, specifically focus on the anticancerous, anti-glycation, and anti-diabetic activities. And lastly, we have also evaluated the biocompatibility of these synthesized silver nanoparticle against the human red blood cell, and also evaluated toxicity against the Brine-Shrimp larvaceae. This is the briefly methodology that is I have discussed in the objective slides. Now coming towards our findings, first we have done the phytochemical analysis in terms of the total phenolic contents and total flavonoid contents and the free radical scavenging activity. As we all know that the plant extracts are naturally enriched in various kinds of phytochemicals, let's suppose like polyphenols, ketones, carbohydrates, and other different kinds of secondary metabolites, which have the natural capability of reducing the metal ions to form the metallic nanoparticles. So when we have performed the TPC and TFC analysis, these plant extracts was found to be rich in the phenolics and flavonoid contents, which were corresponding to their highest free radical scavenging activity. Now, how we synthesize the silver nanoparticles? In case of the silver nanoparticle synthesis from the Morus-Mediated plant extract, first of all, we go for optimization of the exact ratio for the synthesis of the silver nanoparticle, in which the silver nitrate solution, which was used as a metal precursor salt, and the plant extract was mixed in various definite ratios, like into 1:1, 1:2, 1:5, 1:10, 1:15, and 1:20 to optimize or to maximize the yield of the silver nanoparticles. As we can see in this figure, the initial indication of the synthesis of the silver nanoparticle is the change in the color from light brown to reddish brown, and the reddish brown color was prominent in 1:5, 1:10, 1:15, and 1:20 ratios, and the more darker towards the 1:20 ratio. But finally, we have confirmed the synthesis of the silver nanoparticle in all these ratios by using the UV visible spectrophotometry that is shown in the figure B, the spectrophotographs of the these all ratios, and as we can see that the highest peak was observed in the case of the 1:5 ratio, and the other 115 and 120, which shows the greater color change, have shown not any particular peak in the range of 400 to 500 nanometer wavelength, which is the surface plasmon resonance area specified to the silver nanoparticle synthesis, and only the 1215 and 110, these shows the good results, and specifically the 15 shows the best results or best peak in terms of synthesis of silver nanoparticles. Then further, we have choose only the 1:5 ratio for the synthesis of the silver nanoparticles. For all other further experimentations and also for to evaluate the impact of the UVC light, we have choose the 1:5 ratio. Next, we have performed the UV visible spectrophotometry analysis of the monometallic nanoparticles, silver and zinc, which were we used. One was the control, is the control that was into 1:5 ratio, and the B represent the synthesis of the silver nanoparticles under UVC irradiations. Please keep in mind that the UVC irradiation exposure was given for 2 hours during the constant stirring for the synthesis of the zinc oxide nanoparticles, while in case of silver nanoparticles, the exposure time was only 30 minutes. After 30 minutes of the exposure of the UVC light, we have since the distorted or unstabilized synthesis in the nanomaterial of the reaction mixtures visualized by the UV visible spectrophotometer. Same as in case of the silver nanoparticles, the zinc oxide nanoparticle UV spectra also show a specific surface plasmon resonance in the range of 350 to 400 nanometer, which is a particular range of the surface plasmon resonance of the zinc oxide nanoparticles. Similarly, the bimetallic nanoparticles, silver zinc oxide nanoparticles, one was 0.1 into 0.5 and other was the 0.5 by 1. In which the first we have constant silver that was the 0.1 by 0.1, both the silver nitrate solution and zinc acetate was used into 0.1 molar concentration, while in case of the second concentration, the silver remains constant, that was the 0.1 concentration, while the zinc acetate salt was used into 0.5 molar concentrations. And these two different types of synthesized bimetallic nanoparticles were monitored by UV visible spectrophotometry for the confirmation analysis of the synthesis of these bimetallic nanoparticles. And then we can see in the UV visible spectrophotographs of these bimetallic nanoparticles, the particular surface plasmon peaks corresponding to the monometallic silver and also the bimetallic zinc oxide nanoparticles were two major peaks were observed either in case of the control and also in the case of the UVC mediated bimetallic nanoparticles. These nanoparticles were further visualized or further evaluated by the Fourier transform infrared spectrophotometry analysis to find out the possible phytochemicals responsible for the reduction and specifically the capping of these monometallic and bimetallic nanoparticles. This is the FP spectra of the leaf extract of the Morus-Mediated plant extract, which was used as a reducing and capping agents for the synthesis of the nanoparticles. And this is the spectra of the monometallic nanoparticle. A is of control, B UVC, C zinc oxide, and D is for UVC mediated zinc oxide nanoparticles. In all types of the FTR spectra, we can monitor different transmittance peaks in different wave corresponding to different wave numbers. And when we have interpreted these wave numbers, they correlates mainly to the polyphenolics and flavonoids that was present in the plant extract that was responsible for the capping and the reduction of these types of monometallic as well as the same spectra were observed in case of the bimetallic nanoparticles, and they have also the same kind of bimetallic nanoparticle synthesis. The capping of the bimetallic nanoparticles as revealed by the to find out the morphology, specifically the shape and the size of the synthesized monometallic and bimetallic nanoparticles, we have performed the scanning electron microscopy. In scanning electron microscopy, this figure 7 represents the shapes of the monometallic nanoparticle. A is the control in which there was no radiation of the nanoparticles to UVC radiation, while in case of the B, there was the radiation with UVC light. We can see a huge difference between the unirradiated or irradiated UVC radiated silver nanoparticles. In case of the control, there was almost irregular triangular shaped silver nanoparticles, while light, which has a low intensity and low wavelength, has modified or has changed the physiognomes of the controlled nanoparticles into the most dispersed, non-agglomerated and spherical type silver nanoparticles. While in case of zinc oxide nanoparticles, the controlled nanoparticles data shown in the figure C were somewhat ovulated type. But when they were irradiated with the UVC light, their intensity have changed the nanoparticles into some orthorhombic or hexagonal type that was clear in these of the cube shapes silver nanoparticles. These are the images of the scanning electron microscopy analysis of the bimetallic nanoparticles. A represents the control and B represents the UVC radiated in 0.1 into 0.1 concentration, while D and E, C and D represents the UVC mediated and control in 0.1 into 5 ratio. As we can observe that in case of the controls, either A or either in the C, there was a flower shaped zinc oxide silver bimetallic nanoparticles were synthesized. But when they were irradiated with this UVC light, we can see a clear difference between the needle type and the cubic type zinc oxide nanoparticles. When they further analyzed by the STEM EDX analysis, we found that the zinc oxide nanoparticles were in form of a cubes, while the silver elements present in these bimetallic nanoparticles were in the form of the rod shape. These synthesized nanoparticles, the shapes are summarized in this table, like we have discussed in the scanning electron microscopy, that what were the shapes and either there was agglomerations or not, that we have discussed in the SAM analysis. Further, these synthesized monometallic and bimetallic nanoparticles were characterized by the energy dispersive X-rays analysis to find out the elemental composition of these nanoparticles in all monometallic silver A and B and C and D that represents the zinc oxide nanoparticles. We can say that there are specific peaks of the zinc and oxygen in case of zinc oxide nanoparticles, and specific peaks of silver were present in silver nanoparticles, either controlled or either UVC mediated, which further confirm the purity of these nanoparticles synthesized either under controlled or UVC radiations. The same is the scenario in case of the bimetallic nanoparticles. The particular peaks of silver, zinc, and oxygen were observed in all cases, in all ratios, either controlled or either irradiated UVC radiated silver zinc oxide bimetallic nanoparticles. Lastly, these monometallic and bimetallic nanoparticles were characterized by using the XRD diffraction analysis of the bimetallic nanoparticles. In case of monometallic nanoparticles, you can see that A figure represents the very huge raw background noise, and there was not a particular peaks which was showing to unimpurity and also the not defined crystalline nature of the controlled silver nanoparticles. While in case of the UVC mediated silver nanoparticles, no doubt there is a background noise, but there was a clear faces of the silver nanoparticles, which represents the face centered cubic crystalline nature of the silver nanoparticles. While in case of the zinc oxide nanoparticles in control, there was also a background noise to some extent, but in case of UVC mediated zinc oxide nanoparticles, there were very well defined and sharp peaks which represent the hexagonal versatile structure of the zinc oxide nanoparticles. When the bimetallic nanoparticles, silver zinc oxide nanoparticles were characterized by the XRD diffraction analysis, we can see that in both cases, either controlled or either UVC radiations nanoparticles, this shows the particular peaks which correspond to the silver zinc oxide nanoparticles. However, the effect of the silver metal was masked by the zinc oxide nanoparticles. Zinc oxide nanoparticles in case of bimetallic nanoparticles were the more dominating as compared to the silver in case of XRD diffraction analysis. While the main difference between the irradiated and non-irradiated bimetallic nanoparticles shown by XRD data was the intensity, or that you can say the yield. The yield in case of UVC irradiated nanoparticles intensity was higher as compared to the controls. After fully characterization of these nanoparticles by different characterization techniques, we have further evaluated the different biomedical applications of these nanoparticles in terms of antidiabetic, anti-glycation, and also the anticancerous activity. Firstly, we have evaluated the antidiabetic activity in terms of the inhibition of two enzymes, alpha amylase and alpha glucosidase. As we all know that the alpha amylase and alpha glucosidase are involved in the carbohydrate digestion in our body, and if we can inhibit the breakdown of the carbohydrates, we obviously can control the sugar level in our blood. Then by inhibiting these enzymes, we can control the breakdown of the carbohydrate, or can inhibit the breakdown of carbohydrate in our body, and our blood sugar levels can be controlled. So we further check the percentage inhibition of these enzymes, and we sees that the maximum percentage inhibition of the both enzymes was shown by the NP3 nanoparticles, and NP3 nanoparticles were our UVC mediated silver nanoparticles, which shows the highest anti-inhibition activity, might be due to their smaller size and more spherical penetrating shape. While in case of the anti-glycation activities was also evaluated in terms of two parameters. One was the inhibition of the vesperine-like advanced glycated end product inhibition, and secondly was the pentosidine-like advanced glycated end product inhibition. In case of anti-glycation activities, the end glycated products inhibition was also maximum observed in case of parallel silver nanoparticles, either controlled or either UVC mediated silver nanoparticles shows the best results, and similarly the monometallic either silver or either zinc shows the best results in case of antidiabetic and anti-glycation activities as compared to their counterparts bimetallic nanoparticles. Further, we have evaluated the anti-cancerous activity of these synthesized monometallic and bimetallic nanoparticle against the Hep2 cell lines, and the anti-cancerous activity was evaluated in parameters of different parameters. Like first of all, we have checked the viability of the cancer cells in terms of by using the MTD assay, and we can say that the minimum inhibition was observed in case of the NP7, and almost all types of nanoparticles shows less viability, less than under 30% viability as compared to the non-treated cells. But the highest was observed in case of the NP6. NP6 is the bimetallic silver zinc oxide nanoparticles, which shows the highest inhibition, or you can show the lowest viability of the cancer cell, corresponding to almost 20% cell was only viable and 80% was dead. Further, these anti-cancerous activities were evaluated to evaluate or to find out the possible mechanism, which was studied by using the production of the ROS, reactive oxygen species, and reactive nitrogen species. The results of the production of the reactive oxygen and reactive nitrogen species correspond mainly to the cell viability. The maximum production of the ROS or RNS, which causes the oxidative stress to the cancer cells and lead to the apoptosis of the cancer cells, was also observed in the bimetallic nanoparticles NP6 and NP8. Both are the UVC irradiated zinc oxide nanoparticles that were vary in the concentration either 0.1 by 0.1 or either 0.1 by 0.5. The other aspect that was used for the evaluation of the anti-cancerous activity was the find out the mitochondrial membrane potential, and the mitochondrial membrane potential was also specifically highest damage was observed to the cancer cells in case of the NP6 and NP8 kind of nanoparticles. Furthermore, we have also evaluated the anti-cancer activity of the monometallic and bimetallic nanoparticles by evaluating the gene expression of the caspase 3/7 gene expression, and also by observing there the gene activity. This gene expression and the gene activity was also highly observed in case of NP6, which was the bimetallic nanoparticle, as compared to the other either monometallic and bimetallic nanoparticles. So we concluded in case of the anti-cancerous activities, the bimetallic nanoparticles, specifically the UVC radiant nanoparticles, shows the highest cancerous activity due to their needle type shapes and due to smaller size as compared to the other monometallic nanoparticles, and shows the highest anti-cancerous activity. Lastly, we have also evaluated the biocompatibility was evaluated in terms of RBC, human red blood cells hemolysis, and we have observed that the maximum the hemolysis of the red blood cells in terms of percentage was only 3.5%, which is very least. We can say that these are very biocompatible to the human red blood cells. Either these are the monometallic nanoparticles synthesized, or either these are the bimetallic synthesized nanoparticles. Similarly, their toxicity was also evaluated against the Brine-Shrimp larvae assay by using their LC50 value, and the same case was observed. They were less lethal, or minimum toxicity was causing to the larvae, Brine-Shrimp larvae. Therefore, we have concluded that these green synthesized biometallic nanoparticles not only shows the promising biomedical applications in terms of anti-cancerous, anti-diabetic, anti-glycations, but also were highly biocompatible and she's least toxicity. Then finally, we conclude that these nanoparticles can be synthesized from plants, and they UVC radiation can control their size and shape, and these are biocompatible, and further they can be recommended for other in vivo types of biomedical applications. Thank you so much. Any question?
Thanks for your presentation. Now floor is open for the questions, if anyone have.
Yeah, I have a question. A very nice presentation. I was just wondering, did you test these nanoparticles on normal cells, and if they had any impact on apoptosis in non-cancerous cells?
No, we have not tested them for non-cancer cells, but the biocompatible study, like we have tested against the human red blood cells. These are, you can say, that non-cancer cells, and they were very biocompatible to the human red blood cells, but specifically not any other non-cancer cell line was tested